Hi everyone, my name is Mara Burrows. Today is March 18th, 2020, and this is for week 2, lab 2 for my Bio 225 class. So the first exercise is culturing microbes in the broth. I have all my supplies in front of me, my candles, my cultures. I already have a cup of alcohol in which I will be placing the pipette in right over here. So that's there. I am gonna go ahead and start. So what I'm gonna do is I've placed the pipette in a cup of alcohol to make sure that it enters the stem of the pipette, which is done. I am going to go ahead and ignite a candle safely with my lighter. Okay, that's lit right here. Okay, after that, I'm gonna go ahead and label a nutrient broth, which I have right over here. Nutrient broth. We're gonna label this E. coli with a permanent marker. I'm gonna go ahead and place this near the candle as advised. I'm gonna go ahead and start to exhale the alcohol out of the pipette. So now this is sterile. So while holding this, I'm gonna go ahead and pick up the broth and gently remove the cap. As so. And it says for me to pass the lip of the broth tube through the flame of the open candle. So we'll go ahead and do that. Keep in mind I still have my pipette. Okay. And just turn it as I go to sterilize it. And again, in a circular motion. Okay. After doing that, I'm gonna go ahead and place this on my work surface. I'm actually gonna place it on here so that I have something to hold it. And next I'm gonna take the E. coli culture, which is right over here, and do the same.
So I'm gonna go ahead and open this up, and I'm gonna do the same with the candle, being very careful with it.
Okay, so that's done. Next, I am going to pipette 0.25 mLs of the broth into the nutrient tube of the culture vial. So I will be going ahead and putting that in here. These are 5 ml pipettes, so I'll be doing half full. Okay, let's go ahead and do that. Okay, looks like I did that. I'm gonna go ahead and put this back into the alcohol so that it gets sterilized again. And I'm gonna cover this up. And I'm gonna go ahead and shake it until it fully dissolves.
Move this away a little bit.
Okay, as you can see, it looks pretty dissolved to me. So that's that. Next, we are gonna go ahead and do that with the epidermis culture.
As well. So I'm gonna go ahead and take one of these, take a nutrient broth, I'm gonna label this the S. epidermidis nutrient. Okay. I'm going to go ahead and put this over the candle flame. Kill any bacteria. Put this down. And I'm also going to go ahead and open this up. And do the same with this, being very careful. Okay. I have my pipette, which I took. The alcohol is all out, so I will go ahead and grab some of this broth. And I will go ahead and put it in here to help dilute it. Set this in the alcohol again. I'm actually going to go through the flame again just to make sure that it is sterilized, and then I will go ahead and grab my cap, close it up, and shake it until it's dissolved. Okay, as you can see, it is fully dissolved, and this is the S. epidermis.
Next, we are going to be doing the yeast. So I'm going to go ahead and label the other plastic cup clear as yeast. So what I'm going to do is I'm going to open the yeast packet carefully and apply half a teaspoon of the powdered content into an empty cup and a 1/4 cup of the warm tap water until it is dissolved. So I will be right back. Okay everyone, I'm back. So I went ahead and poured half a teaspoon of the yeast into my yeast cup and mixed that in with a 1/4 cup of warm tap water. So I'm just gonna stir it so that everything gets dissolved. So, okay, it's almost fully dissolved. Looks good to me. Have a little bit in the spoon. Okay, so that's good. I am gonna go ahead and set this down. It'll be ready in about 10 minutes when it starts to bubble. In the meantime, what I'm gonna do is go ahead and label my third broth. This we are going to label as S. cerevisiae. Okay. So while that's setting, what I'm going to do is I'm going to go ahead and uncap this. And we are going to go ahead and start the process again of sterilizing it. So I'm going to go ahead and swirl it over the open flame. Be very careful. That way it's sterilized. I am going to wait a little bit and then I will be back once this yeast starts to bubble up. Okay guys, so I already have these all prepared. What I'm going to do is I'm actually going to go ahead and put these cultures into their broth to mix in. So this is the E. coli. Again, I am going to go ahead and sterilize this again just to make sure that it is sterile. Place that down. Same with the E. coli. Make sure it's sterile. Okay. And with the pipette, I am going to go ahead and take this and put it into this broth without touching it. Okay. And place this back in here and let that sanitize. In the meantime, I'm gonna close this. Next, I'm gonna do the same with the SE. So I'm gonna go ahead and close this E. coli broth. Let that kind of set there. Okay, next I'm gonna take the SE, and I'm gonna do the same. So I'll go ahead and sterilize this broth again just to make sure that it's clean. Same with this, sterilize the tips. Okay, I have disinfected my pipette with the alcohol. And I'm gonna go ahead and make sure it's empty. No alcohol is left there. Okay, and then go ahead and grab the bacteria and place it into the broth without touching, and the remainder as well. Dip that back in the alcohol. I'm gonna go ahead and close this, set it aside, and I'm gonna go ahead and close this for now. That is the S. epidermis. Okay, everyone, so my yeast has bubbled up for the most part. I am gonna go ahead and, just for sterilization's sake, relight my candle. I'm gonna go ahead and re-sterilize this broth. I am gonna go ahead, this has been dried off from the alcohol already, and I'm gonna take 0.25 mLs of this. Looks about right there. And I am gonna go ahead and pour it into the broth. Okay, just like that. Re-sterilize it back. And I'm gonna put this dipper in the alcohol and close this. Close this up. Okay. So that concludes experiment 1. I do have 3 of my specimens here, which I will incubate for 48 hours and see how they grow. Thank you. Hi everyone. So it has been 48 hours since the broths have formed. So this is the E. coli bottle, the S. epidermidis, and the S. cerevisiae from the yeast. So today what we're gonna do is we're gonna go ahead and isolate colonies. So I have already formed 3 agar plates, and I've also labeled them, well, I've labeled all 3 plates, 1 for each bacteria. So this is the S. epidermidis. This is the E. coli. And then this is the yeast, S. epi. So what I'm gonna start on now is I have a candle here. I'm gonna start with the E. coli right here. But before we do that, I have my 3 loops soaking in alcohol. I'm gonna go ahead and light this candle carefully. And we are going to get our E. coli and our E. coli agar plate. Open that up. Gonna go ahead and just put this over the fire to help keep it sanitized. Okay. Next, I'm gonna take a loop, shake it off a little bit to ensure that it's been dried, and I'm gonna dip it in the E. coli as so. And swish it around. And I'm gonna take my Petri dish for the E. coli and I'm just gonna zigzag it over on one cubicle. Move that out of the way. So I went ahead and I'm zigzagging it over right like that. Next, I'm gonna put this back. I'm gonna close this tube. I'm gonna blow out the candle. Take that same one that I just dipped in alcohol, let it dry. I've turned my Petri dish already. I am going to go ahead and start from kind of the inner part and swish it around, up and down, all the way to the edges. And then dip it back in, turn my Petri dish, about 20 seconds there. Alcohol. Again, let it dry. Make sure there's no alcohol in there. Shake it around. And again, I'm gonna take it from the inner corner or the mid corner and just Z it out. Dip it back in. And one more time. We're gonna do the same with the other corner. And again, all around. And close that up. Next, I'm gonna go ahead and close this up, set it aside, and then do the same for the other 2. Okay guys, so I finished doing the isolation on all 3 of my Petri dishes. I am gonna go ahead and set it aside, let it incubate for 48 hours, and then we will take a look at it and see how it looks like. Hope you all are having a good day. Thank you.